Welcome to the deep dive. Okay, imagine this scenario. You're maybe on your first clinical rotation in med school. A patient walks in and they're complaining about this unexplained numbness down their arm, maybe, or weakness in a leg. How do you even start to figure that out as a budding clinician? But it really all starts with understanding the body's incredible, really intricate nerve maps. So today we're embarking on a pretty crucial journey through the nervous system. It's foundational stuff for, well, every aspiring healthcare professional, especially you, our listener, diving into your medical journey. We've got a great stack of materials here from Hilary Manton's lecture on dermatomes and segmental innervation. And our mission really is to make this complex material crystal clear, memorable, and really clinically relevant for you.
Indeed. And it's good to think of the nervous system not just as wires, it's more like a dynamic communication network. Understanding its structure, how signals travel, how different pathways interact, it's absolutely crucial. We'll be breaking down the central and peripheral systems, signal flow, and maybe most importantly, how you can differentiate sometimes subtle nerve damage by really understanding the body's own internal maps.
Right, so let's unpack this. We want to give you some mental anchors, maybe some catchy acronyms too, and even some diagnostic thought experiments along the way, things to help you retain and recall this vital information. By the end of this deep dive, you should be able to describe the divisions of the nervous system, really get the difference between spinal nerve roots and remises. That's a key distinction. We'll definitely clarify that. Grasp a simple reflex arc and confidently differentiate between dermatomes and peripheral nerve territories. That last one, that's like your diagnostic superpower.
Yeah.
Seriously. Okay, let's start with the big picture then. The anatomical and functional divisions of the nervous system. Think of it like the ultimate information system for your entire body. The command center, maybe?
Right. Anatomically, it's simpler than you might think at first. Two main parts. First, you've got the central nervous system, the CNS. That's your body's headquarters. The brain, the brainstem, and the spinal cord. This is where, all the major decisions and processing happen.
EHQ. Got it.
Then you have the peripheral nervous system, the PNS. Think of that as the extensive network connecting the CNS to everything else. This includes all the nerve fibers shooting out and also the ganglia, which are like little relay stations. Collections of nerve cell bodies outside the CNS.
Okay, so CNS is command, PNS is the network.
Yeah.
But the PNS isn't just one thing, right? How does it act on commands? How does it know the difference between, say, me deciding to wave my hand and just, keeping my heart beating?
Excellent question. Yeah, the penis gets specialized. Functionally, we split it again. There's a sensory division. This is the information incoming pathway. It diligently brings signals towards the CNS, everything you feel, touch, see, sense.
Okay, incoming data?
And then there's a motor division. That's the command's outgoing pathway. It transmits impulses away from the CNS to effector organs, so muscles or glands, telling them exactly what to do.
And that motor part, that's where the conscious versus unconscious control comes in.
Precisely. The somatic motor division handles your voluntary actions, the movements you consciously control, like deciding to raise your hand right now. But then there's the autonomic nervous system, the ANS, that's completely involuntary. It's your body's autopilot mode. It manages crucial things like your heart rate, digestion, breathing. You don't even think about it.
It's in the background.
Exactly. And the ANS itself further divides into the sympathetic system, that's your fight or flight response, gearing you up for action, and the parasympathetic system, which is your rest and digest response, calming things down.
Wow, okay. So CNS is the main office. PNS is the delivery service, with sensory coming in, motor going out, and that motor service has the conscious somatic branch and the autopilot autonomic branch, which itself splits into sympathetic and parasympathetic.
Really?
Quite a system. And within that PNS, we also have the cranial nerves. 12 pairs, mostly for the head.
That's right, 12 pairs, mostly serving the head and neck. 10 emerge from the brainstem, and 2 directly from the brain itself. But then we have the spinal nerves. And these are really our main focus for today.
Okay, spinal nerves.
Absolutely critical. They form the backbone, literally almost, of much of a body's communication. There are 31 pairs of these segmental nerves, and they emerge from the spinal cord. Most of them are what we call mixed. That means they carry both afferent sensory signals information towards the CNS, like feeling a light touch, and efferent motor signals commands away from the CNS, like telling a muscle to contract. They're multi-talented nerves.
Okay, mixed signals, 31 pairs. Now, building on that, it makes you wonder, if a reflex, like pulling your hand from something hot, is so incredibly fast, where does that initial processing happen? Does the brain always get involved right from the start?
Let's hold that thought. We'll come back to it as we zoom in on the spinal nerve itself. So we've got the big divisions down, and we've introduced these crucial spinal nerves. But how do they actually manage the body's fastest responses? Let's zoom right into a cross-section of a spinal nerve, our information highway. See how those urgent messages get processed. Okay, so in a spinal nerve, right where it connects with the spinal cord, you essentially have two main paths for information flow in the gray matter. The posterior aspect, sometimes called the dorsal horn, that's where sensory information comes in. Think of it like an on-ramp for all the incoming data.
Sensory in the back. Got it.
And the anterior aspect, the ventral horn, that's where motor information goes out. That's your off-ramp for commands. It's a really beautifully clear, organized system for somatic stuff anyway. Sensory in, motor out.
Right. Okay, so that organization helps. Now back to our earlier question. The fast reflex. Where does that processing happen?
Yeah.
You mentioned the simple reflex arc, the body's express lane. How does that work?
Yeah, the reflex arc. It involves sensory neurons, those afferent fibers, bringing information to the CNS. Then motor neurons, the efferent fibers, transmit impulses out to effector organs, telling them to react, like pull your hand away. Now, sometimes in between those two, there's an inner neuron that does a bit of processing. But here's the kicker, and this answers our question. In the simplest somatic circuits, like the classic knee-jerk reflex everyone knows, that sensory neuron synapses directly with the motor neuron in the spinal cord.
Directly. No middleman.
No interneuron needed for the simplest ones. This direct connection completely bypasses the higher brain centers for incredible speed. The brain actually receives the message after the action has already started. It's more like an FYI, this just happened signal to the brain.
Wow, so the brain finds out after the leg kicks. That explains the speed. That's genuinely amazing. It's such an efficient system. Life-saving, really.
Yeah.
Okay, so to help you anchor this, maybe think of the SMART reflex. Sensory in, motor out, afferent fibers, reflex arc, target effector. Smart.
Good one. Now, before we dive into the maps themselves, the dermatomes and myotomes, there's a quick but really important structural point about spinal nerves. We said they emerge from the spinal cord, but what happens right after they emerge? This is where we need to talk about spinal nerve roots versus rami. Understanding this difference is crucial for figuring out where a problem might be located.
Ah, yes, roots versus rami. This was one of our key objectives. And yeah, I remember this tripping up students. Can you break down what rami are? How are they different from the roots we might already be thinking about, the ones coming right off the cord?
Absolutely. Think of the nerve roots as the initial connection. You have a dorsal root carrying sensory info into the cord and a ventral root carrying motor info out of the cord, purely sensory or purely motor at this point.
Okay, dorsal sensory, ventral motor.
These two roots then merge very briefly to form the actual spinal nerve, which is mixed. This short spinal nerve then exits the spine through the intervertebral foramen, and immediately, like right after it exits, the spinal nerve splits into branches. Those branches are the rami. Ramus is just Latin for branch.
Ah, okay, so the roots combine, form the nerve exits, then it branches into rami.
Exactly. And you typically get two main ones. A posterior ramus, which heads backwards to supply the muscles and skin of the back itself, and a much larger anterior ramus, which sweeps forward and out to supply the limbs and the front and sides of the trunk. And the key difference. Both the posterior and anterior rami carry mixed signals, both sensory and motor fibers distributing the nerve's influence, unlike the roots, which were separate sensory or motor pathways initially.
That makes perfect sense. Roots are the specific on-off ramps to the cord. The short nerve is the merge point, and the rami are the mixed distribution roads from the nerve out to the body regions. And this whole segmented setup, it starts way back in embryonic development, right? This pattern is laid down early.
Precisely. In the embryo, you have these structures called somites. They differentiate. Parts of the somite form vertebrae and ribs, but another part, the dermatomyotome, is key here. It further divides into the dermatome, which gives rise to the dermis, the skin layer, and the myotome, which forms specific muscle groups. Trunk and limb muscles mostly come from the anterior ramus supply, back muscles from the posterior ramus. Each of these segments gets its nerve supply right from the start and pretty much keeps it.
So each segment of the body has its own dedicated nerve connection, tracing back to a specific spinal cord level, a built-in map.
Exactly. The CNS is segmented, and each segment connects independently to the periphery. Sensory nerves serve these discrete territories of skin, the dermatomes. Motor nerves serve muscle territories, the myotomes. They retain that original innervation.
Okay, let's get into those crucial maps then. First up, dermatomes. The body's sensory zip codes, you call them.
Yeah, that's a good way to think about it. A dermatome is specifically the area of skin supplied by the sensory fibers of a single spinal nerve root. So, for example, the C6 dermatome is a specific strip of skin running down your arm and into your thumb, roughly. Now, there can be some overlap between adjacent dermatomes, which is sort of a safety buffer. If one nerve is slightly damaged, you might not lose all sensation there. But there are also very distinct boundaries called axial lines. These are junctions where dermatomes from spinal levels that are not next to each other meet up, like C6 and T1 on the arm. There's no overlap there. It's a sharp line.
Gotcha. And these dermatome maps are incredibly useful clinically, right? For pinpointing spinal cord or nerve root issues. You mentioned anchors earlier. These are great for the trunk dermatomes. Really helps you remember them in practice. Let's recap those. T4 is roughly at the level of the nipples. Think T4 at the nipple floor. T6 is around the xiphisternum, the bottom tip of your breastbone. T10 is at the umbilicus, the belly button. That's the classic TEN to the belly BUTTON. Hard to forget that one. And T12 is down at the pubic bone level. Having those landmarks makes assessments much faster.
Definitely. And alongside the sensory map, the dermatomes, we have the motor map, the myotomes. A myotome is a group of muscles innervated by the motor fibers of a single spinal nerve root. Just like testing sensation in a dermatome tells you about the sensory root, testing muscle strength or reflexes associated with a myotome tells you about the motor root. For example, testing the biceps reflex checks the integrity of spinal levels C5 and C6, while the triceps reflex checks C7 and C8.
Sensory map, motor map, dermatomes, myotomes. Makes sense. Now, a quick but really important practical point you touched on about the spinal cord itself. It doesn't run the whole length of the spine.
Correct. That's a common misconception initially. The spinal cord proper actually ends around the level of the L1 vertebra in adults. It's shorter than the vertebral column. Below L1, the remaining spinal nerves have to travel downwards within the vertebral canal before they can exit at their correct level. This bundle of descending nerve roots looks kind of like a horse's tail, so it's called the cauda equina.
The horse's tail. Right. And also the way the nerves exit. You said there's a rule except in the neck.
Yeah, generally spinal nerves C8 through the rest exit below their correspondingly numbered vertebra. So the T5 nerve exits below the T5 vertebra. But in the cervical spine, C1 to C7, the nerves exit above their numbered vertebra. And C8 exits below C7 and above T1. It's just a quirk of how things line up there.
Good detail to remember. Okay, here's that crucial thought experiment again for you, our listeners, our future diagnosticians. Patient comes in. Numbness on their arm. How can you tell? Just by mapping out where the numbness is. How can you tell if the problem is up near the spine, affecting a spinal nerve root? Or if it's further down the arm, affecting a peripheral nerve branch? This distinction is absolutely key. It's your diagnostic superpower, and we're about to unlock how to use it.
Right. This distinction is probably one of the most clinically relevant things for a first-year student to really, really nail down. It fundamentally changes where you look for the lesion, the source of the problem. Remember how nerves grow into the limbs during development? They don't just run straight. They form these complex networks called plexuses, like the brachial plexus in the arm or the lumbosacral plexus for the leg. These are like intricate interchanges where fibers from multiple spinal nerve roots get mixed and reorganized.
So they interweave, like braiding ropes together.
Exactly like braiding ropes. It's a fantastic safety feature. If you damage just one single spinal nerve root contributing to the plexus, you usually don't lose all function in the limb. The other roots contributing fibers via different routes can compensate. It provides redundancy.
That's brilliant. Makes the system much more robust. Okay, so the plexuses mix things up.
They do. And then emerging from these plexuses, you get the named peripheral nerves that we all learn, like the radial nerve, the median nerve, the ulnar nerve in the arm, or the sciatic nerve in the leg. Now, a peripheral nerve territory is the specific area of skin innervated by one of these named peripheral nerves. And here's the crucial part. Because of the plexus mixing things up, each named peripheral nerve contains fibers that originated from several different spinal roots.
Okay, I think I see where this is going. That mixing is the key to differentiating.
Precisely. This is where the answer to our thought experiment lies. This is where your clinical detective work kicks in. Let's break it down. A dermatome, remember, is the skin area supplied by a single spinal nerve root. Neurons from that one root might actually travel out through multiple peripheral nerves eventually. So if you see sensory loss that fits a dermatome pattern perfectly, it points to a problem at the root level, close to the spinal cord.
Like a pinched nerve root from a herniated disc.
Exactly like that. That's a classic root problem, or radiculopathy. Now, a peripheral nerve territory is the skin area supplied by a specific named cutaneous nerve that came out of a plexus. This nerve carries fibers from multiple spinal roots. So if the sensory loss follows the distinct map of a named peripheral nerve, it points to a problem with that specific branch nerve somewhere after the plexus, distal to the spine.
So the pattern of numbness is the giveaway. Is it following a single root's distribution, dermatomal root problem, or is it following a named nerve's territory, peripheral pattern branch problem?
You've got it. That difference in the pattern is your massive clue. It literally maps the lesion for you. Think about it like troubleshooting electricity in a house. If a whole circuit is out in multiple rooms, maybe you check the main fuse box, right? That's like your spinal root lesion affecting a dermatome. But if only one specific outlet in one room isn't working, you look at that outlet or the wiring in that room. That's like your peripheral nerve lesion.
Great analogy. So if a patient has numbness in, say, the classic C6 dermatome pattern down the arm to the thumb, you're thinking nerve root compression at C6 level.
Correct. You'd investigate the neck, the cervical spine.
But if they have numbness just in the pinky and half of the ring finger, in that specific pattern,
Then you're thinking ulnar nerve. That's a peripheral nerve territory. The problem is likely further down the arm, maybe at the elbow where the ulnar nerve is vulnerable, not necessarily back at the spine. Or carpal tunnel syndrome affecting the median nerve gives a different, very specific pattern in the hand.
or that regimental badge area numbness on the shoulder.
Classic axillary nerve territory. Again, a peripheral nerve issue, maybe from a shoulder dislocation, not usually a root problem.
Okay, this is really clicking. Let's simplify it even further. Maybe a quick reference.
Good idea. Let's summarize the key differences. Spinal segmental nerve root. Location, near the spinal cord. Origin, directly from a spinal cord segment. Fiber content, fibers from one spinal segment. Clinical use, identifying spinal nerve root lesions like radiculopathy. Peripheral nerve, location, distal in the body, out in the limbs usually. Origin, forms after plexus formation. Fiber content, fibers from multiple spinal segments. Clinical use, identifying peripheral nerve injuries or entrapments like carpal tunnel or a nerve laceration.
That's super clear. Spinal root, one segment near spine, peripheral or nerve, multiple segments mixed, further out. That distinction really is a cornerstone of neurological diagnosis, isn't it?
Absolutely fundamental. Mastering this allows you to approach neurological symptoms logically and efficiently.
What an incredible deep dive that was. From the big picture of CNS and PNS down through the roots and rami, the reflex arcs, to this really intricate dance of dermatomes and peripheral nerve territories. We've covered a lot of ground, and hopefully this provides an invaluable foundation for, well, for your entire medical journey ahead.
Absolutely. And remember, understanding these patterns isn't just, academic hoop jumping. It's profoundly essential for your future clinical practice. Being able to accurately map out sensory deficits or motor weakness in a myotomal pattern allows you to precisely locate nerve damage. And that guides accurate diagnosis and ultimately more effective treatment for your patients.
So don't forget those SMART reflexes. Remember the trunk dermatome landmarks T4, T6, T10, T12. Use those anchors. And especially, really burn into your memory that critical difference between dermatomal and peripheral nerve distribution. That's your diagnostic superpower we talked about. These aren't just facts to memorize, they are tools. Tools for your neurological exams for figuring out what's going on.
Well said. And maybe here's a final provocative thought for you to consider as you continue your studies and eventually move into practice. How might this precise mapping we've discussed, understanding dermatomes, myotomes, peripheral territories, how might that influence future advancements? Think about pain management, perhaps developing highly targeted interventions based on these maps, or even nerve regeneration therapies. If we can map the loss so precisely, maybe we can better target the repair. It really opens up a world of possibilities down the line.
That's a great thought to chew on. The future applications are fascinating. Thank you so much for joining us on this deep dive. Keep exploring these pathways, keep learning, and we'll catch you next time.